Hello and welcome to the ConTech Live Weekly webinar. I'm joined today by Dale Morgan, who is Senior Account Manager in Wiley's Global Sales Partnership team, and Vatsal Agarwal, Vice President, Artificial Intelligence and Computational Linguistics for Innoplexus. And Vatsal is based in Frankfurt in Germany. The subject today is that Dale and Vatsal are going to talk about using AI to enhance speed and precision of biomarkers discovery. Highly specialist, very specific, and very interesting in and around the AI space. Just a few words of introduction. It's a Zoom webinar, therefore your mics are all muted, but you can file questions using the Q&A tab. Please do, and we will wrap up all the questions at the end of the session. That's pretty much all from me. We're going to run for 20-25 minutes and then 5 to 10 minutes of chat and wrap about 5 p.m. BST. So all that remains is for me to hand you over to Vatsal and Dale.
Thanks, Clive. Yeah, I'll pick up initially. So all I'm going to do is really provide a quick bit of context, a little bit of background before we dig into Vatsal's more interesting component. But let's just get this up. There we go. So yeah, you know, many of us will have seen a lot of headlines that look a bit like this one in the last few months about the role that AI is playing in the race for a vaccine against COVID-19. And you know, what I think is that we're seeing the acceleration because of the urgency with which we need a vaccine, you know, real acceleration of the number of changes that were already taking place. And those are changes around workflows, of course, and that's around the use of data and the application of machine learning to problems across a variety of healthcare use cases, whether it's drug discovery, clinical trial design, precision medicine, as we will get to. And you know, additionally, I think we're starting to see it even in the business development and marketing areas. But these are all you know, behind the AI banner, specific use cases, all of which require their own use of data. But those changes, I think, are beyond the lab somewhat as well. And I think we're seeing changes in the ways that companies collaborate, both in the sense of the ways that they access data and are able to use it, and you know, also around the production of insights from those data and the ways that companies in collaboration are acting on them. So it's a use of data driven by urgency, but that data-led approach poses its own challenges which we will get into. So while there's a real great need to stay focused on a core problem, there are obstacles that need to be looked at in order to efficiently exploit the data that's available. And in that sense, I think COVID is a mirror of a general trend. You know, the use of machine learning solving approach based on data, but at a time when the data available is really growing rapidly. The question of whether you can get that data into enough. That data can take, you know, many forms. It's companies' own internally produced data, and to that, you know, they might want to add publicly available data from sources like PubMed, clinicaltrials.gov, and increasingly, you know, a greater number of open access literature and accompanying data sets that go with it. And on top of that, you know, they might be choosing to add additional data, proprietary content that might be added under a license or in partnership, whether that's published literature or, you know, from a healthcare institution, you know, to really grow the variety and completeness of data that's available. So can that be added to the pipeline while allowing, you know, the core company to remain focused on its main job and its main business? And currently that's proving a challenge. Look at how data scientists can spend their time 4 days out of 5 on average that, you know, they might not be producing insights. It can take on average a week to gather data that comes from a variety of sources and in disparate formats coming from many locations, and an additional 3 days to clean and normalize that data, which, once other things are taken into account, only really leaves half a day for really mining for patterns in that data and start to extract insights, which really highlights the need to have a really clear strategy and culture around data. And we're starting to see an increase in the number of data scientists that pharma companies have, who need the right data, complete, accessibly stored, and geared towards the right kinds of insights. So, I mean, setting a clear strategy around data, and also having a data culture that ensures the core business is really focused on the right kind of insights. And on top of that, I think partnership strategy is starting to become part of that as well. And we're seeing new relationships appear again around finding vaccines for COVID-19 that are acceleration of the kinds of things we've seen before. Ways to access different or better technology that a company might not have in-house, ways of sharing expertise, and to hear new perspectives on familiar problems and new problems at the same time. All of which kind of points us towards one specific use case that we're going to look at today, which is looking at precision medicine and the discovery of biomarkers, which Vatsal is going to talk about from the Innoplexus perspective. Add up, and here we go. So
thanks, Dale. There are a few topics I would like to cover, but I think we can start with the basics first. So what is a biomarker and how does it help in precision medicine? Dale already highlighted a few points about the availability of data, but data scientists spending 80% of their time on fetching it or cleaning it rather than actually solving the problem. Now, biomarkers is one of the problems. Many of the AI companies are trying to solve it. But what is that problem? First of all, biomarker is generally a biological marker. Anything biological which can mark a state of a biology of a person, anything measurable, which can relate to an outcome in a much more quantifiable manner. The most common ones or the popular ones are the omics data sets. Genomics, proteomics, metabolomics, and all of those, they are the more popular ones because that's where you have wealth of information. But to understand simply, there can be many other things. They can be from your lab tests. How much is your hemoglobin level? How much is your RBC and WBC counts, etc. That numbers that you get. It can be as simple as temperature. If your body temperature is high, you have fever, you need to give something in combination to help reduce that. Or your blood pressure and heart rate and all of these things which we don't really see as a fancy biomarker, they can help differentiate a condition, a state of body, and how to deal with it. So these are the number of kind of things you see: physiological, demographic. Same thing which might work for men may not work for women. It may work for a younger population, but we see for COVID-19, a lot of the things are impacting more on the older population. So can we differentiate and can we build things specifically? That's what biomarkers enables us to do. But how does it help in precision medicine? So if you have any biological marker or a combination of them, it can help you in diagnosing the disease. Many of the diseases, for example, let's say very simply migraine. How many people in the world have migraine? Millions of them on a daily basis. But till date, we do not know how to kind of quantify migraine. Or how do we observe or how do we differentiate between two people having migraine? Is it the same thing or a different thing? So these biomarkers, if we can find a biomarker for migraine, that this person having this kind of a feature is different from the rest of them, and this person can be treated using these therapies. That's what biomarker enables us. It helps us set every individual apart in the diagnosis of the disease, prognosis of how it would proceed or the severity of the disease, response of specific drugs, for example, chemotherapy. How many people get chemotherapy and it's only working in a certain percentage? Say in best case for some therapy, it might be 70 to 80%. But still, those 20 to 30% people are going through immense trouble of chemotherapy without it working at all. So can we identify those people and segregate them much earlier? That's the promise of biomarkers and precision medicine, which we are trying to solve using AI. The next section is more about what challenges. So if really biomarkers can solve the problem, then why do we not have them? And I would bucket them into 4 categories. The first is availability of data. Some data sets are available much easily. So you can subscribe to Wiley journals, to any of the journals and publication houses, and you get that data. A lot of that is publicly available in the open source databases. These are ENCODE and 1000 Genome and GEO, and everybody has a lot of human data. But a lot of people are also keeping it private. So first, finding the right set of data is a challenge that is applicable for the use case. And second is the heterogeneity. The few examples I gave. So GEO has a different format from what ENCODE has or from different data types. It's very heterogeneous. Especially, did they run the experiment with the same parameters? Probably not. Everybody would have their own assumptions and own border cases of what they included and what they excluded, which uniquely defines that data set and not comparable to any other. So how you homogenize these disparate data sets is another problem. The third bigger problem is the complexity of human biology. In usual cases, we have test data from hundreds of patients in a best case scenario. So you have basically 100 outcomes or few hundred outcomes. Whereas in each body, there are hundreds and thousands of different potential markers. We have 20,000 plus genes, 100,000 plus proteins, and then all of the physiological differences. So how can 100 or 500 patients define the 10,000 parameters? That's the complexity level that we cannot. And the final part is the dynamic nature. The values of these proteins and genes in the body is different in evening than morning, is different in China, different in Europe, different in US, different in males, different in females. Their eating habits, their lifestyles, everything changes. So there is a lack of transparent mechanism that I can say these are the reasons. If I get higher value of a patient, doesn't mean it would be reflected in anybody else because it's caused by a number of other parameters. So these are the complexity. First two around being able to handle that much data and use it in which AI can solve, but also the complexity of human biology and transparency of being able to find that. And to control that, again, AI can help us, which is what I'll be covering in the 2 sections. One is speeding up the data preparation. The first 2 problems of data, you have data from many published journals I mentioned, and the NGS databases. Enterprises have their own data. So somebody who is working on Alzheimer's would have the data from Alzheimer's patient, which is not publicly available, but it needs to combine with the public data. And the collaboration data. Why is the data from other departments in the same company not being leveraged? So currently, it's a difficult challenge to assimilate the data within the same company across departments or across prior researches to find that out. And your partners, you are working with a lot of laboratories, a lot of data generators. So Handling all of that is a complex issue. One thing which enables us is, or we would think is a mandatory requirement, is a conceptual search. Instead of just looking, I think Dale also briefly mentioned, looking at keywords or searching for COVID-19 related things, it may be written in 100 different ways. There might be indirect references. So how do you identify a paper related to COVID-19? And then you have further constraints on this kind of population, on these kind of things. You need a search engine and an algorithm which can search and annotate the concepts which are specific to life sciences, which means something in life sciences, and you are not just doing a string search. An interesting example here for ADHD. So although ADHD is fairly common in the medical community that it's a disease, but there is also accidentally a protein which also happened to be known as ADHD, and an equivalent gene which is ADHD1. So I don't want to mix these results. These are different results and should be treated differently. And that is where the first step, finding the data, available data from different sources, can come into the play. Once that data is there, then what we call, funnily enough, is data mess. It's like you have all sorts of data from the world, but it's not usable. If you see on the left side, this is what you get despite the best effort, despite making it in the best, easiest manner. This is what the data looks like. It's annotated, it's using some of the standardizations, and still getting in these few formats I've mentioned. There are 10 more. And somebody might be calling a disease condition in a different word, severity in different metrics, gene and protein names from different databases. And what that would do is 100 data sets, meaning 100 times the effort for each database to bring it into standard form. This is where the life sciences language processing I was talking about previous slide helps us bring it into the same format. Now you see here the Crohn's disease way. It may be called by any other way in the actual data with pipe separation, etc. But in the output, it is shown as clearly a disease, which can then be used further. And similarly for every other thing. And extending, so this is actual use case we did using data from Wiley, was these publications have a lot of tables which are wealth of information. But somebody has to sit and read each of these articles to find the right biomarker or at least their activity in the human body. Again, the same engine using computer vision, another one of AI technologies, can extract these pieces into such organized forms. So you know what is the biomarker and all of its properties in a tabular manner, which machine can then analyze. So this is the first problem that we focused on, getting the data and putting it into these automated pipes. Humans put in the hard work. A generic comment, everybody seems to think AI is a magic button, which gives you the final result. Here is the biomarker. We kind of differ. AI can help in a lot of lower cognitive tasks. Finding the data, combining it, removing the batch effects and all of those. But the actual insights, we need the humans, as of now at least, to decide what is the right insight if machine gives them all the landscape in the consumable form. So this is what it does, taking each of the data sets, putting it into tabular formats, and then combining them, because each experiment has a different assumption. So this is a real case where we ran. You can see each color represents one experiment. So each experiment is very separately distributed than other experiments. So you run some of the standard AI algorithms to combine them into an overlapping set, that they should all have now the same assumption, the underlying effects have been removed. But still, as you see the purple set here, it doesn't comply no matter what we did. Now we don't want to overfit, we don't want to necessarily fit something which is not in the right constraint, so that we can skip. So this is the proof of all the similar talking experiments coming together, whereas something which is different, let's take that separately. After that is the key goal. Once you have prepared all of that data in the right form, then you pass into AI-based models to see on different hierarchical clustering parameters which genes go together, which are in patient segment A versus B versus C. Now these segments can be low severity and high severity, responding to a particular drug or not responding to a particular drug, healthy or diseased, basically any segment. This way, biomarker will be able to differentiate between patients. But as you remember, I mentioned there are hundreds and thousands of different parameters and only few hundred patients. So you would get more likely false positives, the biomarkers which are not really biomarkers. So that's why where we go into our proprietary technology of combining all of them biologically. That if we look at all of these biomarkers, how their biology is, how connected they are, then some of them come on the very periphery of it. That means this particular gene, protein, or RBC count, something is not related to anything else in this process. How is that possible? It's more likely an offset because of some other parameters or other processes. We would like to ignore those or give them very low weight and combine them with or look at more the clusters within. The central clusters is where we would like to focus. And then on the right side, you have the list of biomarkers which are part of the core cluster. So this kind of analysis, more zoomed in, you pick up a cluster and then you run Monte Carlo simulations. It's called Monte Carlo simulations because as in Monte Carlo gambling, you see your odds in 21, what's the odd of you winning and so on. We run millions of simulations that if this gene is the biomarker changing this, what would happen? So not just looking at that specific biomarker, but entire network, are the other effects observed in the literature, in the publications and all of that wealth of knowledge that this is a good biomarker. That's the key element. Getting the data in, then finding the candidates which are differentiated but may not be causal, and then looking in the entire connected biology, what is causal, that can then be given some confidence. And this whole workflow coming from hypothesis generation to the suggested subgroups of patients, in all of these steps, it brings in the transparency. Usually, if I go within this box, only the computational modeling, it's very difficult to say why something is there. And people have this constant complaint that AI doesn't give you the answer, or it doesn't explain why something is so. But because of this process of validations and connecting through the publications and literature out there, you can say that this gene affects this pathway, and this pathway has a role in this disease, and this is why this might be an alternative biomarker, or this might be, you can generate your clinical findings. So AI helps you at all of these steps: aggregating data, computational modeling, networks, and finally scoring and validating on the literature. And ultimately, a researcher wants to see bunch of nice graphs and very key publications or literature rather than what data scientist does. So this is where we kind of visualize all our outputs in a platform that researchers can just go in, study a particular biomarker, its literature, its network, and they say this makes sense for our drug or our medicine, and we would take this up to test clinically. That's all I had prepared, and I think we can open up the floor for questions.
Hi. Thank you so much, Vatsal and Dale. That was a pretty much perfect example of a ConTech issue where we see the power of AI and brand new technologies impacting on the way scientific content is utilized. I love the reflection that you don't get all the answers, and one of our keynotes famously refers to AI as augmented intelligence, not necessarily purely artificial intelligence. Another good example of that. And congratulations, you're the first webinar presenter that managed to get Monaco into a presentation. Just looking at questions, I have one very specifically about the methodologies and things. A wide-ranging question: how is it different from what the industry has been doing for the past 2 decades?
So to that, I would say that there have been number of methodologies people are trying. The first step of data aggregation, data finding is not different from what industry is doing, but it is speeding it up. Instead of person going and searching every single thing on a PubMed, on Wiley, and then pulling it together, machine can do that much faster and much effectively. And the second differentiator would be that network simulation. The biggest challenge in biomarker industry has been that I find a biomarker which doesn't correlate clinically. So when I was doing AI, it was coming up good, but when I go to clinic, it doesn't work. So when we combine the literature evidence and do those network analysis, then it gives much higher chances of success than what has been tested so far.
Excellent. Okay. We all are aware that things are moving very, very fast, and we are dealing with leading edge technologies. How is this theoretical, or have you actually tested all of this in practice?
So these things, some of the graphics, some of the images I showed you are actually from the project that we have done. We have now tested it in 4 different scenarios in 4 different therapeutic areas. So we can say, I wouldn't say it's perfect yet. We are still moving towards that. But being proven in 4 different therapeutic areas for 4 different clients, it's definitely showing promise of making things easier and faster for them.
That's fantastic. We are very close to time with just a couple of minutes left. So thank you, Dale. Thank you, Vatsal. That was a really terrific presentation with a lot of detail. I'm pleased to say that everyone who's attended will have access to a completely transcribed and indexed video of the presentation. That should be available tomorrow. We use a company called Zeotag, which is also AI driven to actually process those. So that will be in the wider public domain soon. One more webinar scheduled for next week at 4:30, the usual time. We are discussing making better science easier. And that's being presented by Dr. Leslie McIntosh, who is the CEO of Ripita. Again, a very, very ConTech live relevant topic, I hope. So if you haven't registered for that already, please do. Thanks once again to Dale and Vatsal. All I need to do now is wish everyone a good evening, good afternoon, and we hope to see you all again on the ConTech Live circuit very soon. Thanks. Bye-bye.
Thanks, Clive. Bye-bye.
Clive.